Hello everyone, my name is Kritika from MSc first semester. Today we will discuss about Plasmodium, the malarial parasite. First of all, we'll discuss about its life cycle of Plasmodium vivax, which causes malaria. Plasmodium vivax, it is cosmopolitan in distribution. It has two hosts, a vertebrate host in which reproduction is asexual, that is intermediate host, human, and a blood-sucking insect in which sexual reproduction takes place, mosquito, that is definitive host, and the vector is Anopheles mosquito. It comprises of three main phases, schizogony, gamogony and sporogony. Schizogony has two phases, exoerythrocytic that takes place in liver cell and erythrocytic schizogony that takes place in RBCs. In exoerythrocytic schizogony, infection occurs when mosquito bites human, and at this stage parasite is called as sporozoite.
Sporozoite has a slender, slightly curved, sickle-like body tapering at either end. It is only 15 micrometer long. It is enclosed in a thin, elastic but firm cuticle which maintains its shape. The broad middle part of the body has a nucleus, which is the middle part of the body, and the sporozoite is capable of slight gliding and vibrating movement. Sporozoite circulates in the blood for about half an hour after their entry. Then sporozoites leaves the blood and enter into the parenchymal cells of the liver. They remain there for about 12 days and the sporozoites become spherical or rounded shape that is known as cryptozoite. Now cryptozoites feeds on cytoplasm and grows in size into a large rounded structure, the pre-erythrocytic schizont. Later, they undergo multiple fission and develops into about 1000 tiny parasites called as cryptomerozoites in a schizont. Schizont and the liver cells break up and liberate the cryptomerozoites into the liver sinusoids. Cryptozoites, after its entry into a fresh liver cell, is known as metacryptozoite. Here it grows in size and metacryptoschizont is formed. Later it undergoes multiple fission and it forms about 6 to 1000 of parasites called metacryptomerozoites. These are known as metacryptomerozoites, which later on are of two types. One is macrocryptomerozoites and another one is microcryptomerozoites. Macrocryptomerozoites are larger in size and micrometacryptomerozoites are smaller in size. The above described schizogonic cycle occurs in the liver cells before the parasite invade the red blood corpuscles. Therefore, it is known as pre-erythrocytic schizogony. It takes about 12 days. Thereafter, the parasite follows the line of action. The smaller ones or micrometacryptomerozoites enter the RBCs. Smaller ones are micrometacryptozoites, which enters the RBCs and start their erythrocytic schizogony. And the larger one re-enters the fresh liver cells and again restarts the post-erythrocytic phase. Inside the red blood corpuscle, the parasites become rounded and grows, feeding on the cytoplasm of the corpuscle. During the growth period, it is known as trophozoite. The trophozoites develops in the body a large vacuole which pushes the nucleus to one side. This makes the trophozoites looks like a signet ring stage. Further growth, the vacuole disappears and the trophozoites become irregular, and this is known as amoeboid stage. With the help of its pseudopodia, the trophozoites ingest the cytoplasm of the corpuscle. The cytoplasm of the corpuscle is digested and the hemoglobin split into globin and hematin. Of cytoplasm gathers around each daughter nucleus so that 12 to 24 tiny uninucleate bodies are formed. These are known as merozoites, and the merozoites arranged themselves around the residual cytoplasm like the petals of a rose flower. That stage is known as rosette stage. After 48 hours of the infection, the weakened corpuscles ruptures and releases merozoites into the plasma along with the residual cytoplasm, hematin, and toxins. Residual cytoplasm, hematin are eaten up by special cells called as phagocytes in the spleen, kidney, liver. Merozoites in the RBCs do not return to the liver cells. Instead, develops into compact rounded bodies which lacks a vacuole and grows more slowly, and they are known as gametocytes or gametes. There are two types of gametes which are formed during this phase. First one is female gametocytes, second one is male gametocytes. Male gametocytes are also known as microgametocytes, which are smaller or fewer, whereas female gametocytes are larger and more numerous. Gametocytes are unable to develop further inside the human blood due to the high body temperature of the host, so they die if not sucked up by the female Anopheles mosquito in a day or so. The mosquito sucks along with the blood not only the gametocytes but also the stages of erythrocytic schizogony. The stomach of female Anopheles mosquito, all the stages except the gametocytes are digested. They break corpuscle in the stomach of the mosquito and become free. Development of the gametocytes in the mosquito's stomach involves two important events, that is gametogenesis and fertilization. Gametogenesis means formation of gametes. The male gamete correspond to spermatozoa. Its nucleus divides to form 4 to 8 daughter nuclei which migrates to the periphery of the cytoplasm. A bit of cytoplasm gather around each daughter nuclei. The male gamete are attached to the unused cytoplasm of the gametocyte having hematin in it. Soon they become free and start creeping by lashing movement of their flagella in the search of female gamete. Female gamete correspond to ova. Its nucleus casts off into one or two small pieces which leave the gametocyte with a bit of cytoplasm. This converts macrogametocytes to macrogamete, correspond to polar bodies formed in female gametes, develop into a small projection, the cone of reception on one side. One male gamete penetrates the female gamete through the cone of reception. The nuclei and cytoplasm of both the gametes fuse to produce a single cell, the zygote. Syngamy involving the fusion of dissimilar gametes is called anisogamy or heterogamy. Zygote becomes elongated and worm-like and starts moving on lining of stomach. It is now called as ookinete or vermicule. The ookinete passes through the wall of the stomach just beneath its outermost layer. Here it assumes a spherical form and gets enclosed in a thin elastic permeable cyst wall around it. The cyst wall is derived partly from the zygote and partly from the insect tissues. At this stage, the parasite is known as oocyst or sporont. Oocysts look like a rounded projection on the surface of stomach. There may be 50 to 500 oocytes on the stomach of the infected mosquito. Now, sporogony. The oocyst grows in size, drawing nourishment from the mosquito through the permeable cyst wall. The cyst wall also enlarges to accommodate parasite in it. The cytoplasm of the oocyst develops a number of vacuoles and its nucleus repeatedly to form a large number of daughter nuclei. Each daughter nuclei surrounds by a bit of cytoplasm. The resulting bodies are called sporozoites. Sporozoites mature to form spindle-shaped forms. Oocyst membrane rupture releases sporozoites in the body. Majority of them enters the salivary glands and wait for the transfer into the
blood of man with the mosquito bite. Entire development of parasites inside the mosquito takes about 10 days to 3 weeks depending upon the temperature. The sporozoite again enter by the bite of mosquito inside the human blood and the cycle repeats again. So take a look of life cycle of malarial parasite. Now pathogenesis. Commonly involved organs are changes in blood, spleen, liver, bone marrow. And signs and symptoms are like abrupt fever, chills, rigors, headache. Initially may be continuous, then periodic, up to 41 degrees centigrade or 106 degree Fahrenheit temperature, nausea, vomiting, abdominal pain, well between febrile episodes, splenomegaly, hepatomegaly, anemia, falciparum fetal because of brain and kidney damage. Laboratory diagnosis includes thick and thin smear tests. Other laboratory diagnosis includes blood examination, TLC and DLC. In TLC, if low TLC, it causes leukopenia. Other diagnosis includes biopsy, therapeutic tests, serological tests like fluorescent antibody test, complement fixation test, flocculation test, hematogglutination tests. Preventions include chemoprophylaxis, mosquito netting, window screens, mosquito repellent, protective clothing, special care during night time, DDT or kerosene oil spray over pools of water, drainage of stagnant water, and no vaccine is presently available for malaria. That's all. Thank you.